Okay.
Okay, good afternoon, everybody. Thank you for joining today's webinar. We're going to go ahead and get started. My name is Stephanie Lemon. I'm Professor in Chief of Preventive and Behavioral Medicine at the University of Massachusetts Medical School. This is the second webinar coordinated by the Community Engagement Cores of the Clinical and Translational Science Institutes at the 4 medical schools in Massachusetts: UMass Medical School, Tufts Medical Center, Boston University and Boston Medical Center, and Harvard. The Clinical and Translational Science Institutes are funded by the National Center for Advancing Translational Science at the National Institutes of Health to academic medical centers in order to transform the research enterprise to make larger impacts on human health. The community engagement cores of the CTSIs in Massachusetts have a consortium to work together on common priority areas. We decided to put this webinar series together as a way to share the research going on at our institutions related to COVID-19 with our community partners, other stakeholders, our academic colleagues at other institutions, and across our own campuses. We're very excited to have such a large and diverse group of participants today who represent industry, government, nonprofit, clinical care, academia, and others from Massachusetts across the country, as well as several other countries. At all 4 of our institutions, much like medical schools and other institutions of higher academic learning across the country, our research enterprises have really transformed in the wake of COVID-19. From lab-based research to community research, investigators have quickly shifted the focus of their research to address the many aspects of this disease and the complex medical and social consequences of the pandemic. We are very fortunate, and I am privileged to be able to introduce today's speaker. Dr. Anastasia Khvorova from the University of Massachusetts Medical School is a professor in the RNA Therapeutics Institute at UMass. She'll be talking to us today about the work that she is leading. The title of her talk is RNA Therapeutics, Informational Drugs as a Pandemic Response Tool. To orient you to our agenda, we'll move to the next slide. Dr. Khvorova will be talking for about a half an hour. That will be followed by a question and answer session. To orient you to how that is going to work, we are going to hold all questions until the end, but we're going to utilize the Q&A function of Zoom. If you look on your screens at the bottom in the middle, you can see where it says Q&A. Please feel free to enter your questions into that box. At the end of her presentation, we will have somebody read the questions to her, and we will ask questions in that manner. At the conclusion, we're going to have a quick wrap up, and I'm going to tell you a little bit about some of the next steps for our seminar series. So with that, I would like to turn it over to Dr. Khvorova. Thank you so much.
Thank you, Stephanie, for a great introduction. And I'm going to share my screen. So what my lab does normally at UMass Medical School, it's a lab working on chemical biology of therapeutic RNAs. Essentially, we're trying to understand how the chemical architectures of the siRNAs, guide strands, antisense, define the ability to silence Gs in vivo and become a drug. And the lab is located on a 4th floor of this building. So when COVID-19 happened back in January, several people in the lab essentially approached me and said, you know, Anastasia, we have an ability to deliver to the lung. We have an ability to make a hyperfunctional compounds. Let's make a drug because we have an ability to do it very quickly. And what I will be talking today to you today is giving a general description of what it takes to make a COVID-19 or antiviral response drug in a very short period of time in a context of academia. So we all know that the SARS-CoV-2, which now have been sequenced extensively, is a virus which is causing this COVID-19 pandemic, which we're living as we speak. And it's very similar in structure to some other coronaviruses, and it has a complex genome with 9 open reading frame and multiple mRNAs. There's a diverse clinical manifestation for this virus. People with healthy immune systems are not developing significant symptoms or minimal amount of symptoms, and some people get severely sick. And essentially the concept behind the treatment is that if we would be able to reduce the viral load significantly enough to allow the adaptive immune response system to develop the antibodies and actually get rid of the infections, this is a very viable path forward for both therapeutic as well as preventive care for COVID-19. So my lab is mostly working on this new class of molecules, which is called siRNAs, and they were originally discovered within UMass Medical School, also by another professor at the RNA Therapeutics Institute, Craig Mello, which got a Nobel Prize back in 2006, together with Andrew Fire, for discovery of this fundamental mechanism called RNA interference. Essentially, what it means that if you take double-stranded small siRNAs and introduce it into the cells, they load into this complex and serve like a Google of the cells, finding the targets based on sequence homology, cleave those targets and prevent the expression. So oligonucleotide therapeutics and siRNAs in particular are informational drugs. What it means is that PK/PD behavior is fully defined by chemical scaffolds which drive in distribution. And the major difference from conventional drugs is that in the context of a conventional drug, the pharmacophore, so it's a combination of chemical features which is required to recognize a target, is interlinked with dynaphore, which is a combination of features which are required to drive the delivery and acceptable toxicity profile. So it takes many, 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 many years to get those compounds which will have acceptable activity as well as acceptable ADME-tox profile. With oligonucleotide therapeutics, it's a completely different picture. So as long as you have a chemical architecture which gives you robust delivery for a tissue of interest optimized, you can easily reprogram it with different sequences and essentially silence them in a tissue of interest. And what we're doing right now, we are doing exactly that, reprogramming previously developed chemical architectures which support long delivery with the sequences targeting SARS-CoV-2 as well as several other targets, host targets involved in both infection as well as exaggerated immune response. So siRNA therapeutics, as many other disruptive technologies, follow this typical hype curve. So when it was discovered in 1998, everybody got excited. It's such a powerful mechanism. It's going to silence every single gene on demand. In 2006, Craig Mello and Andrew Fire got the Nobel Prize. Big pharmas enter the market with the famous Merck SiRNA acquisition deal for $1.1 billion. And then things are being rushed in clinic where the chemistry is not optimized. Actually, those dynaphores are not optimized. And naturally, they all fail or they show very limited clinical utility. And then majority of big pharma are scared. They're leaving the field. It's never going to work and there's a huge disappointment. But what's happening when Big pharma get disappointed. It means that chemists have to work more, and indeed there is a lot of very talented chemistry work which happens in the last decade, which resulted in a first drug approved in 2018, and right now a panel of close to 50 drugs advanced towards late stages of clinical development. The major transformative discovery in the context of RNAi-based therapeutics was discovery that conjugation of fully chemically stabilized siRNAs, there's not a single ribose left, it's fully chemically stabilized, to multivalent GalNAc result in extremely long duration of effect, where single sub-Q injection, just think about it, single sub-Q injection give you up to a year duration of effect. So at this point, this concept has been realized for liver, and recently there is a data published from the several phase 3 clinical trials from a drug called Khvorova, which is targeting PCSK9, where they have shown up to a year duration of effect of clinical efficacy. And this was developed together by a medicine company and Alnylam. But I think the most fascinating part of this technology is that while this half a year to a year exaggerated duration of effect is demonstrated, it's demonstrated at effectively clean adverse event profiles. There's nothing reported with exception of small fraction of patients developing minor and resolvable injection site reaction. So this drug was recently acquired through the medicine company by Novartis, I think for $9.7 billion, which give you an idea of the power of oligonucleotide and siRNA therapeutics. The key, of course, is delivery, because clinically right now the delivery is optimized only for one tissue. It's lung, it's liver, and also there are certain things in clinical delivery, clinical development for CNS, and my group actually contribute to discovery and optimization of chemical architectures for CNS delivery. The concept we had for pandemic response program, my lab, as well as a very close collaborator, John Watt's lab, who is another lab at the RNA Therapeutics Institute next door, have been working for many years on optimizing chemical scaffolds, which promote delivery to extrahepatic tissues. And in particular, we have several chemical scaffolds. It's essentially the same fully chemically stabilized siRNAs, but they're conjugated to entities which support productive lung delivery. And the idea was, let's identify lead compounds targeting SARS-CoV-2, put it in a chemical scaffold which gives you lung delivery, and see whether this can be used as something for prevention or treatment or prophylactic of COVID-19 infection. So what do we need to have actually to make this happen? We have to have fully chemically modified scaffolds, because this full chemical stabilization is absolutely essential for multi-months duration of effect in vivo. And we are targeting, we want to see this 2 to 4 months duration of effect, because to some degree this can be used as a treatment, but maybe it can be also used as a very quick, short-living vaccination concept for people at high risk or people with a high degree of potential viral exposure. Second most important thing, we have to have chemical architectures which support efficient lung delivery with acceptable safety profile, and I will talk about it. The third one is actually informatics of siRNA design for targeting viruses is pretty complex, because on top of favoring the features which are defining ability of those very complex chemical architectures to interact with risk complex and silence genes in vivo, one need to ensure that we're targeting regions of conservations and potentially targeting the regions of conservations not only within multiple patients with the same type of virus, but potentially targeting other coronaviruses. So in the future, when future pandemics happens, you can maybe introduce minor modifications in a drug entity and have a ready-to-respond drug. One need to identify lead compounds efficiently targeting SARS-CoV-2 as well as several other host targets. And what is very important, establish experimental system which allowing you targeting and evaluation and impact on SARS-CoV-2 infection both in living cells as well as animal models. So chemistry is fundamental for development of oligonucleotide therapeutic drugs. And what is very, very important in the context of RNAi, we have to introduce those modifications, which gives us stability, duration of effect, promote from excessive innate immune response activation, and promote lung delivery, but do it in a way which wouldn't interfere with the compound's ability to efficiently interact with the cellular machinery. And this is the key balance which need to be kept in place. So in clinic right now, there's 4 major type of chemical modifications being used, which is phosphatide modification of the backbone, 2'-O-methyl 2' modification of the sugar, and optionally, it's 5' stabilization of the phosphate, and it can be a vinyl phosphonate, and there is a lot of publications about it, but there's a variety of other modifications which can serve the same function. So I just want to emphasize again that this full chemical stabilization is absolutely essential for observing robust delivery to lung and robust delivery to many other tissues. And without full chemical stabilization, extrahepatic exposure in particular is very, very minimal. Just to give you an idea that if you compare partially modified siRNAs versus fully modified siRNAs, and you look at the degree of tissue exposure and accumulation with the same dose levels, it's black and white. Like even having a single unmodified ribonucleotide, it's sufficient to block long-term tissue accumulation. What is important to understand as well is that how you modify the sequences makes a huge difference. And there's a very interesting story which happens in the field of RNAi is that when Alnylam moved ahead with the first GalNAc conjugated drug, rivuzaran, which was active in vivo, active in patients, but it's need to be administered weekly at 200 milligram dose. So the total yearly exposure of the patient was around 25 grams, and it wasn't very stable. And this particular drug failed in phase 3 clinical trial, and it failed because of significant mortality. And etiology of this observed significant tox have never been resolved. On top of it, this inclisiran, which is a very similar compounds, and what I'm showing here, it's 2 chemical configurations which give you an idea of what they are. And if you can, and the inclisiran, this is a compound where single sub-Q injection give you almost a year duration of effect with no reportable adversible events, with exception of injection site reaction observed after dosing of thousands of patients. And the difference on a chemical level between those 2 chemical configurations is a couple of extra phosphatide backbone modifications and a couple of extra methyl modifications. So it looks minor, but it has profound impact on both efficacy and safety, and duration of effect. So what we have been working a lot in a lab, and we developed a wide variety of novel chemical scaffolds, which has ability to enter into this complex the same as a traditionally modified compound, but have substantially orders of magnitude enhanced stability. And what I'm showing here is just three chemical architectures. One is ribosoram-like, another one is inclisiran-like, and novel scaffolds we use in the lab, and you can appreciate that we're gaining an orders of magnitude in stability, and this translates in better tissue accumulation and potentially a longer duration of effect. So if I look at our summary slide, absolutely, we do have chemical scaffolds which support multi-month efficacy in vivo. Okay, so what about the lung delivery? There's two ways how compounds and oligos can be delivered to the lung. One is systemic, and the beauty of systemic delivery is that it's very easy clinical development. It's single sub-Q and IV injection. It's uniform lung delivery because you're delivering through the blood flow, through capillary infrastructure. There's no impact of patient clinical condition or lung inflammation, and specifically with advanced COVID-19 patients, their ability, like the lung function can be compromised, and ability to deliver locally can really change patient to patient. But the negative impact of this mode of administration is there's clearly significant exposure to other organs, which might be good and bad. It's just part of reality. With local delivery, which is usually delivered through just nebulization of approximately 1.5 microliter of liquid solution of the oligo, and there's many commercially available devices where the droplet size is optimized to ensure uniform distribution through the lung. And this supports delivery not only to the lung, but to a respiratory tract, which is in initial site of the SARS infection. But it can be affected by patient clinical stage. Nebulization, specifically in a context of an active COVID-19 infection in clinic, provide certain restrictions on how it can be administered, because you are potentially increasing the chance for the host proficients to get infected, but it allows you very selective silencing in the lung with pretty much minimal systemic exposure. So how do you really develop oligos which will give you a robust delivery to lung, rather systemically or locally? And this is not easy. And unfortunately, I only have half an hour, so I cannot go into details of exact years of chemical evolution which got us to this point, but I would like you to understand several concepts. So when you think about optimizing oligonucleotide chemistry, you're effectively playing Lego. You have to modify the rate of clearance, which is absolutely essential because non-conjugated, non-modified oligonucleotides, neither with local delivery nor with systemic delivery, would not reside in the tissue and will be cleared from the body very quickly. You have to have those advanced chemical stabilization patterns, and it makes a huge difference. And both the structure and how you modify the oligo have an impact on delivery. Actually, the chemistry of the linker makes a huge impact because it modifies the ability of oligo to escape from endosomes. And of course, the chemistry of the conjugate itself makes a huge difference. So while GalNAc, multivalent GalNAcs deliver to liver extremely well, they do not deliver effectively to any other tissues. And other class of compounds, which is hydrophobically modified oligos, have been explored for a while because they are supporting this more uniform and delivery to a wide range of tissues. And the conjugate, which is widely used and published extensively, is cholesterol. And while, yes, it allows you to deliver to multiple tissues, and in this case, the red color represents RNA, siRNA present in a tissue because it's fluorescently labeled. It has significant toxicity in vivo, at least in the context of fully modified compounds, so its therapeutic use is limited. So what we did, we decided to explore the chemical space of wide range of different conjugates and see whether we can identify compounds with a better efficacy, safety, delivery ratios. And in particular, we look at different fatty acids, different head modification groups, steroids, vitamins, non-hydrophobic entities, as well as looking at the different valency of those configurations. To test the impact of the entity of the conjugate on the delivery, we usually perform a simple experiment where we take the mice, we inject, and we look at distribution by combination of fluorescent microscopy, PMA hybridization assay, which is assay it's not dependent on presence of fluorescent label, and then we'll look at the efficacy studies looking at the target knockdown. So what you see here is a summary slide, and in this particular case, we're looking at the panel of different conjugates and just showing you that by changing the identity of the conjugate, and the only thing which is changed here is identity of a conjugate of the siRNAs, we can completely shift kidney liver distribution profile, where the more hydrophobic compounds preferentially goes to the liver, while less hydrophobic compounds cleared more and retained more in the kidney. So what is driving this dramatic shift in distribution profiles? And the predominant effect is how the nature of conjugate changed the ability of the oligo to bind to the serum proteins. And practically what you see, if you see a heavily hydrophobically modified oligonucleotides, they mostly bind to LDL and follow the LDL distribution profile. Less hydrophobic binds HDL and follows HDL distribution profile. And then certain types binds albumin, which gives you a more diverse variety. So what we have done after the screening of many different conjugates, we have identified several scaffolds, and you probably can see this black and gray bar here, which clearly gives a much better delivery to the lung compared to everything else. And this delivery is safe, at least with single injection. We have a variety of portfolio of data in non-human primates shows that, yes, you get lung delivery and pretty much no detectable adverse events. Just as a comparison, if you look at the lung here, if you compare, in this case, it's a conjugate called DCA, compare it to cholesterol, which is previously used, you almost get an order of magnitude better lung accumulation. Of course, the most important question is whether the sufficient lung accumulation translates in efficacy, and the long story short, absolutely yes. And now we have a portfolio of different chemical scaffolds, which gives us robust delivery in multiple tissues. And what is relevant for COVID-19 project is we have robust delivery and silencing in the lung. So as I started this presentation, there are two ways to deliver, systemic and local. And what is important to understand, the chemical architectures which gives you local delivery are completely different than from chemical architectures which give you systemic delivery. If you try to locally administer lipophilically modified oligo, it's a no-go because you will get a dramatic inflammatory response. So those two delivery projects are essentially, and screening projects are happening in parallel. But what we were able right now to identify is several chemical configurations which gives us either robust delivery to lung locally. So in this case, it's intratracheal administration. And what you see is just lung of the mice injected, not injected, but treated with PBS, treated with siRNA without a conjugate. So there's a little bit of delivery, but it's very minor. And then siRNAs in a context of this local delivery conjugate, and you can appreciate you get a really robust and uniform distribution throughout the lung, where we confirm uptake practically in all cell types. The same, but with a different chemical architectures, we now have optimized several chemical entities, which gives us very good functional delivery to lung, but systemically. And as I mentioned, there might be different, while the chemistries are different, their APA, so the guide strand of those sequences is identical. It's just what conjugate you attach will be different depending on the route of administration. Another very interesting advantage of this differential route of administration is potential for other tissue exposure. So in a gray shadow is data for intratracheal administration. So we're nebulizing the lung. So we're delivering it locally to the lung, and conjugate 1 and 2 have been delivered systemically. And then when you look at the lung, siRNA by itself, without the conjugate, does not give you significant silencing. Then this local conjugate give you robust silencing, as well as several systemic conjugates give you robust silencing. But what is beautiful is that when we're doing local delivery with conjugate 3, it's in red. We get lung silencing and effectively no activity with other tissues. And when we're looking at the conjugate 1, we do get lung silencing, comparable lung silencing, but we also get activity in several other tissues. So it's really will need to be defined clinically which routes is better, and we're working for it. And it's quite possible that it's not that one is better than another. It might be different targets in different contexts and advance of disease. It's possible that like local administration might be preferred as a prophylactic of infection, because the duration of effect of those compounds is expected to be several months. And in this case, the lung is not inflamed. There is no disease yet, so you're preventing the disease from development. It also might be beneficial for targeting certain host genes where the systemic modulation is not an option. For example, ACE2 is a very interesting target, and we will be talking about it, but you cannot silencing in heart and muscle because of the adverse events. But if you can demonstrate the selective silencing in the lung, it might be a path forward. With more systemic delivery, where you're getting silencing and efficacy in multiple tissues, it may be more a better route of administration for more advanced disease. But at this point, we have optimized chemical scaffolds, which give you robust delivery to the lung, both with systemic administration, local administration, and we are getting this actually 3 lead conjugates in place, which gives us very nice and reproducible lung silence. Okay, so we know how to modify the oligos. We know how to deliver it to lung with different chemical architectures. How do we apply this towards the COVID-19? So the SARS-CoV-2 virus has around 30 kilobase, and it has 9 open reading frames, and it's actually it's an RNA virus, so it's being mutated all the time. So what we were doing at the time of initiation of this project, we downloaded the sequence information available in CBA, and at the time there was 718 patient variants. I think right now it's over 2,000. But we align all of them and making sure that when we're selecting regions for targeting, we're only selecting regions for targeting which has more than 95% conservation. On top of it, there are many other coronaviruses, and while you cannot identify a single 16 base pair region, which is required for productive siRNA targeting, which will be conservative in all coronaviruses, we favor the regions where the degree of conservation is high. So, for example, instead of like going, this is how majority of the sequence space look like, but you can identify certain target sites, which has a higher conservation ratio. So in the long term for the future pandemics, if you have a panel of those compounds, they might be much easier to convert with minimal sequence changes to compounds which will be active against novel type of viruses. Another very, very important factor is bioinformatics of designs, because you essentially have 30,000 different options. And even with our synthetic capability, screening 30,000 will take a while. So what we do, we develop bioinformatic algorithms which give us the ability to predict functionality of the sequences and combine that, and currently the best predictive power we get with machine learning approaches, but we combine that with looking at the high conservation and essentially get the panel of candidates. So we are very lucky because one of the things UMass Medical School has invested into and RNA Therapeutics Institute has invested into, it's Nucleic Acid Chemistry Center, where we have more than 10 different synthesizers ranging from Dr. Oligo 48, where we can do thousands of compounds a week, to Oligopilot 100, where we can make large-scale material, which is sufficient to support non-human primates and large animal models. So what we did, we synthesized design compounds, make artificial viral reporters, which is a lot of effort, and then screen all of them in those reporters. And now we have at least 2 lead candidates targeting SARS-CoV-2 against all 9 key regions. So when you are targeting RNA virus, and this concept is very well understood in the oligonucleotide field, is in general targeting the viral space, one compound is not enough, because if you're just targeting one region, you have a chance of escape mutant, and also from the alignment of different viral genomes, there is a chance that it's not going to be targeting all of them because it's constantly mutating. So what we're doing is at this point, we are using a cocktail of 3 siRNA compounds, and there's different configuration in place. We have our lead configuration where we feel that we're simultaneously targeting plus strand virus as well as multiple secondary mRNAs, which gives a most effective way to go after it. But in the future, it can be something else, or it can be also a cocktail of oligos in conjunction of targeting host targets. Okay. So we have fully chemically modified scaffolds, lung delivering scaffolds, advanced bioinformatics, and we actually do have the lead compounds in place. So what is important to understand as a general design concept? It's target conservation, multiplexing, and multiplexing, cocktail as a current lead, and the reason why oligonucleotide therapeutics is beautifully mapped or developed for that is because the PK/PD behavior is defined by the chemical scaffolds rather than sequence itself, which positions this technology platform as ideal platform for drug multiplexing. On top of it, FDA actually defines a cocktail of the several siRNAs drugs as a single drug entity. Okay, so while I started the discussion with targeting SARS-CoV-2 directly, and this is what we're doing first, because I think from a development perspective, after talking to many people involved in clinic, this is a better path forward. There is less concern of unintended toxicity, but targeting host factors is another very interesting concept, and there's several of them which have been already identified in the SARS-CoV-2 infection. The one which everybody talk about is ACE2, and the ACE2 is expressed in the lung epithelia, and interaction between spike protein and ACE2, this is what absolutely defining the initial stages of infection. And in the context of ACE2, having an ability of only targeting lung and nothing else, but potentially gives us a competitive advantage over small molecules and antibodies, which efficacy cannot be tissue specific. Another very interesting factor which is understood about biology of SARS is the fact that the S1 receptor, upon binding, it's need to be cleaved, and this cleavage allows membrane fusion. And the SARS-CoV-2 does not produce a protease that cleaves its S protein. There's actually several papers which potentially indicate that this acquisition of those 4 amino acids, which everybody talks about it in the context of SARS-CoV-2, generating the site which allows the furin, which is another widely expressed protein, to cleave it and promote an infection. And it might again an interesting target for siRNA going after with siRNAs. There is several others like TM, PRSS2. Right now, if you open Nature Science, it's so many new interesting biology talking about the viral pathogenesis. And essentially, as long as you have a chemical architectures which give you robust and safe delivery, they can be very quickly adapted to go after the therapy. So at this point, our concept has evolved towards not only targeting the SARS itself, but also targeting host factors involved in initial infection or potentially involved in exaggerated immune response. And again, ability to do it locally makes a huge difference. So what's next? It takes a village to move things forward, and we currently have ongoing collaboration with Rob Finberg lab, who famous infection scientist, and he is running a huge lab working on influenza here in UMass. And we're doing testing of the multiple lead compounds and cocktails in a context of infected cells. We also have entered collaboration with Anthony Griffins from BU School of Medicine, who has been running and studying different non-human primates and different animal models of infected with SARS-CoV-2 as well as other viruses, but in a context of BCL3 and BCL4 facilities. And yes, it's New England, but now it's a different university, it's BU. So what we believe is that in a very short period of time, and of course it's years of prior work, and a significant investment in existing infrastructure, we were able to fulfill all the requirements necessary to move the compounds very quickly towards therapeutics. So what we have right now, we do have the systemic delivery to lung. We have chemical scaffolds for local delivery to lung. Platform in place to synthesize and screen and predict clinical quality siRNAs and lead compounds against SARS-CoV-2, as well as several host factors potentially important for COVID-19 disease progression. The major outstanding questions for us is we get lung silencing, but it's not 100%, and we want to make sure that this is sufficient to have a clinically relevant outcome. With systemic delivery, we have oligo exposed to many different cell types. And while we know that it's safe in healthy non-human primates, we want to ensure that we're not creating additional trouble in a context of disease animals. We have a lot of internal discussions and external discussions with clinicians. What is the best time of treatment and what is the best route of administration? And probably it's like with the pickles, it might be not necessarily one solution. It's probably depends on a clinical manifestation and use, and what might be optimal for prophylactics may be a different cocktail and different route of administration for treatment of severely affected patients. At this point, compounds targeting COVID-19 can go through emergency use authorization, and this is what our plan is, and FDA has really clearly defined their expectations and what need to be in place to move this forward. With that, I just wanted to give you an idea that it's with the current advances to oligonucleotides as a technology platform, there is a clear possibility, which we now have executed for SARS-CoV-2, to get from the sequence of the virus to potentially compound, hopefully moving into patients within a very short periods of time of 4 to 6 months, which would be completely unheard of in a context of conventional drug development pipelines used with small molecules and biologics. With that, I would like to emphasize that it takes a village of people, and the data I presented today is a collaboration of multiple labs, and in particular, we're working very closely with John Watt's lab. Our offices are next door, so it's very easy. But he has developed a platform which supports intratracheal delivery to the lung. We also have ongoing collaboration with our animal facility, and without their help, working under very stressful environment right now, nothing would be happening. It's another collaboration with Bob Finberg to do live viral cell testing, and Anthony Griffins from BU for actually non-human primate testing of infected animals. And with that, I also have to acknowledge, not have to, I have an honor of acknowledge a team of people in my lab who are absolutely amazing. You know, last 3 months make me believe in humankind, because the level of dedication, hard work, cooperativity, which the lab and our collaborators has displayed, it's completely unprecedented. And thank you for your attention.
So
Thank you.
Thank you so much for that. We're going to turn now to question and answer time. We're going to use the Q&A box. We've got several questions in there as well. If you have any additional questions, please write them in. And actually, Mary Beth from Dr. Khvorova's lab is going to read the questions today. So Mary Beth, are you ready?
I am.
Terrific.
I'm going to start with a couple questions from early in the presentation, Anastasia. How can modifications to stabilize siRNA affect the RNA-induced silencing complexes, binding, or the degradation capacity?
I actually believe it's both, because advanced stabilization, the reason why we get those exaggerated duration of effect is that all oligonucleotides are being internalized by the cell through endocytosis. And then after internalization, they're stuck in endosomes and lysosomes. And unless they're chemically stabilized, they're degraded. When they're chemically stabilized, they're slowly released from those endosomes, which serves as an intracellular depot for continuing release of the oligo. And this is when this extensive chemical stabilization start to have a profound impact on duration of effect. The more stability, the longer duration of effect. On top of it, we have several cumulatively data suggesting that the half-life of loaded Swiss complex with optimally modified chemically modified siRNAs also enhances the half-life of RNP, which also translates in better potency and duration of effect. And I just have to emphasize that when you're delivering to liver, where it's primary filtering tissues, high blood flow, fenestrated endothelia, stability matters, and it makes a huge difference. And we have seen this rivuzaran and inclisiran story. But when you're delivering extrahepatically for lung, this impact of stability is potentially even more pronounced.
Okay, a couple more clinical questions now. What cell types in the lung will be targeted by a COV therapy?
So this is more, because of the time, I decided to exclude those slides, but this is a result from a collaboration with John Watt's lab, who developed amazing assays which allow you to essentially look at what cell types we get delivery to and individually look at different cell types where you're getting silencing. So clearly different chemical scaffolds give you slightly different delivery, but so far we have data indicating that we get delivery to endothelial cells, epithelial cells, to some degree residual immune cells, and fibroblasts, very good delivery locally, not that good delivery to fibroblasts systemically. But I think the most important cell type for us is endothelial, epithelial cells and endothelial cells, because those are the primary cells which transfer infection.
Do you have any ideas about getting the drug to the lungs if the lungs are like in the setting of pulmonary vasoconstriction?
Precisely, and this is why I was talking about difference between systemic versus local delivery. It's quite possible, most likely, that in a context of severely diseased patients, intratracheal delivery is not an option. And right now, after talking to clinicians, while we get equally good distribution with sub-Q and IV administration, we actually decided to favor IV administration to go forward for those kind of advanced patients. And the reason for that is that with IV, you know you get the full injected dose, and then essentially patient clinical status and potentially affected PK/PD from sub-Q administration is not going to be a point of concern. But it's exactly why we are essentially in parallel developing both routes of administration, because intratracheal delivery for advanced patients probably is not going to be feasible.
Does the duration of silencing require continuous expression of the target? Like, will SARS-CoV-2 siRNA delivered prophylactically hang around for months in the absence of its target to the same extent of siRNA targeting a host gene?
Our current belief that the duration of effect is an exposure to the cell is not affected by the target expression, and we have not seen differences in duration of effect on a impact or whether the target is present or not. And again, think about the mechanism. So it's endothelial uptake, so it's endosomal uptake, and then there is this intracellular depot of entrapped oligos which are being continuously released and loads of Swiss complex. So essentially it's provide to some degree short-term, not long-term vaccination, but it's short-term RNAi-based immunity against a viral infection. And what we're doing, we're just reprogramming the natural mechanism, because RNAi naturally evolved in like in C. elegans and Drosophila, even in humans, as a response to viral infections. So the only thing we do, we're adopting is a natural mechanism of this antiviral response to fine-tune it towards response to the virus, which is a current pandemic.
And how long did it take to design and screen the lead siRNA for COVID-19?
So I will tell the story that actually when you are dealing with 718 patient variants, it's bioinformatically, it's a very intense task, and there is no commercial software which can do it. So the student in the lab, her name is Katie Monopoly. So around middle of the day, we kind of make a decision that we're doing it. So she wrote the code and a software, and she's a very good bioinformatician and computer science student, but she wrote the code, download the data, run the algorithms, run the designs, and probably she was done by like 5 o'clock in the morning, and by next morning, this type of the designs have been on a synthesizer when the lead compounds are being synthesized for screening. This is exactly to emphasize the level of dedication students and postdocs and like everybody, clinicians, show for this project is completely unprecedented. For the screening itself, we have a capability, and again, people are working around the clock, and I will name somebody else, like I caught Dimas Echeverri, who is our oligonucleotide chemist, coming to UMass at 2 o'clock in the morning to set up the machine, so it's running around the clock. To ensure that we get those compounds synthesized as quickly as possible. So it's overnight to run the design. It's probably 5 to 7 days to synthesize the compounds and QC them, but it was years of developing infrastructure in place. The longest part for us was developing and validating the assays and those artificial viral reporter constructs.
Do we have time for another one or two? Okay.
Yes, we can take one more.
Okay. Can designing siRNAs in regions conserved across related viruses be problematic? It's definitely a problem for diagnosis.
It's a huge problem, and I can tell you right away, there is not a single 16-mer which will be fully conservative between all related viruses. So the viruses do mutate a lot. The reason why we decided to favor that is that in the future, if you have future pandemics, and I think it's absolutely clear right now that we will be having future pandemics, if you are favoring going after the regions with a high degree of conservation, you have a chance that instead of redoing the whole screening process from scratch, you would be able to take existing validated compounds, just modify a couple of bases, and hopefully maintain activity and define new specificity.
All right, so thank you, Anastasia, for your time and your energy in this talk today, but more importantly for this just really amazing and exceptional work that you're leading with your team at UMass. It's really been a privilege to have you on this call. Thank you so much. I've learned a lot, and I'm just really fascinated by everything you've talked about today. Thank you. I'd also like to thank everybody for taking the time.
Pleasure.
No problem. I thank everybody for taking the time to be on the webinar today. As I mentioned, this is part of a series. We are going to be having our next talk on Monday. So Karen, if you could bring up the slide. The next talk next week is going to be hosted by the Harvard Catalyst. You can see on the slide here that Yonatan Grad is going to be talking. The title of his talk is Getting to the Post-Pandemic Era, Herd Immunity, Social Distancing, and Vaccine. Just two final concluding thoughts. One is that I think we may have had a couple of questions that went unanswered. We have the ability to take any lingering questions that you have. Please email your questions to communityengagement@umassmed.edu, and we will make sure that Anastasia gets them and that we get back to you. Also, if you have any additional comments or thoughts on this, please email us with your questions. We will make a recording of today's talk available to you. There will be a follow-up email after the presentation today that provides the information on how to access that. So with that, I would like to conclude today's just fantastic presentation. Thank you again, Anastasia, and thank you again to everybody who helped organize and coordinate this. And I appreciate everybody's time and attention. Thank you.
Thank you.